Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond. So today we're going to be talking about storing data in DNA. And I think I came across this topic when I was watching this Netflix documentary series called The Future of, which is quite interesting, where they talk about different things like urban design or death and how it will be like in the future. And this problem of data storage came to light, and I had never thought about it before, although I am very aware of, like, I think the most underrated thing in the modern world is the fact that the cloud, like the data cloud, is not a cloud. Like it's not a virtual thing. It's a physical thing that takes up space, giant warehouses that cost millions of dollars to maintain are storing your data, which is what we think of as the cloud. And on Earth right now, there exists about 10 trillion gigabytes of digital data, and another 2.5 million gigabytes will be produced daily as we write emails, take photos, send out tweets. All of this is creating data. And traditionally, long-term solutions for storing this data is mainly by using hard disks or magnetic tape, where digital data is all encoded in binary code. So it's a series of zeros and ones in different forms. And that's how all of our data is stored, which is also crazy to think about. And yeah, these data, like I said, is stored in these giant facilities, which are known as exabyte data centers, because 1 exabyte of data is equal to 1 million gigabytes. And nowadays, because we're just constantly creating so much data, people are really looking for new ways to store this data efficiently that can be more sustainable in the long term, because like I mentioned, it's taking up a lot of money, electricity, and it's just not very sustainable. So people have started to turn to inorganic materials, polymers, and most recently, DNA, which is a natural form of data storage. And yeah, I find the concept really cool, so I want to talk about it today. So to begin, I wanted to overview how data is stored in DNA naturally for any of our listeners who maybe don't have a science background. So DNA is made up of these chemical building blocks, which are called nucleotides. And nucleotides contain a phosphate group, a sugar group, and then one of 4 nitrogen bases. And this can be cytosine, thymine, adenine, or guanine. And the whole genetic code is encoded by these 4 bases. So for short, we call it A for adenine, C for cytosine, T for thymine, and G for guanine. So it's these 4 letters, A, C, T, and G, that make up the code. So I mentioned digital data is stored in binary form, which is 0 or 1. So there's 2 varieties of the code, but for DNA, there's 4, A, C, T, or G. And then in a strand of DNA, these nucleotides are linked together in a chain, and then 2 strands are joined by pairing of the bases. So A and T pair together, and C and G pair together. And then this makes it a double strand, which twists into a helix, which is the classical DNA helix structure. So DNA actually encodes for proteins. The way this works is that every 3 bases make up a codon, and each codon encodes an amino acid, and proteins are made up of amino acids. So you have a sequence of bases, and then every 3 bases equals 1 amino acid, and then together you can make a chain of amino acids, which is equal to a protein. And a gene is just a sequence of DNA, and that can encode a specific protein. So you can have like a gene for
cortisol.
Yes, cortisol. So obviously, DNA has this high intrinsic capacity for storing information at a very high density. It's actually incredible how high density it is compared to what we as humans can make with our giant data centers. So DNA is 1,000-fold denser than even flash memory. And also, once you make it, it doesn't consume any energy to maintain. It just exists. And so it can essentially be stored forever. DNA can encode 2 bits of information per nucleotide, which makes it theoretically capable of storing 455 exabytes per gram of DNA. So an exabyte of data within DNA could actually fit in the palm of your hand, which would be so convenient if we could have that instead of these giant data centers.
Absolutely.
Yeah, there's been many research groups around the world and studies that have over time been improving the technology for storing data in DNA. And what has really helped with this is because genetics is such a sought after field anyways, and sequencing genes and synthesizing DNA is such a useful thing for so many applications. That's the benefit of this field, that a lot of effort and money and funding and time has been put into improving the technologies. And so now we have much better technology for synthesizing DNA, sequencing DNA, and this is exactly what we need done to encode the data in the DNA, because you need to be able to synthesize it to have the data there. And then you need to be able to retrieve data when you want to. And for this, you need to sequence the DNA and then be able to read it, essentially. And you need to amplify it, which I'm going to go into more later. But there was a study in 2012 called Next Generation Digital Information Storage in DNA. And this was the first time that they were able to encode 1 bit of data per base, like A, C, G, and T, like I mentioned. And they converted an HTML coded draft of a book, which contained 53,000 words, 11 JPEG images, and 1 JavaScript program. And they encoded it into 55,159 nucleotide long oligonucleotides, like strands of nucleotides, basically, that each encoded a data block. And something else is there are a lot of technical challenges. Like, you need to not only have the data itself, but you need to have some flanking sequences that can be useful for the sequencing and reading of the data. And you need to have some extra DNA sequences to label the data so that you can retrieve it. Otherwise, it's essentially useless if you just have the data in this form if you can't read it. But they were able to encode the data, then read the book by amplifying the library and sequencing it, and they could recover all of the blocks. There were some errors because errors are normal, but there were only 10-bit errors out of 5.27 million bits. So it's a pretty high success rate for this purpose.
Yeah, that's really impressive. Because I read one of the issues with errors is that when it's binary code, if there's a mistake, it's binary. So it's either 0 or 1. Whereas with DNA, there's A, T, G, and C. And so when there is an error with DNA, it's usually either insertions or deletions. So if you want to print out an A and nothing appears, it could either cause a deletion. So instead of printing out A, T, G, and C, just print out G, T, G, and C, or insertion. So you go to press an A and it accidentally types 3 A's.
Yeah. And the problem with that, which is also a problem with the genetic code in general, is it causes massive downstream effects. Because in this case, you're not encoding proteins, but normally in the genome, if you had this insertion or deletion, it would influence the whole downstream genetic code. So then all the codons, those 3 base pairs, would change. So then all your proteins, your amino acids, would change. So then your protein will be mutated, which is how mutations work. So I think it really highlights with biology, with genetics and DNA, there's always so much variation, and the error rate is higher than just normal binary digital data storage. So this is something that's not too easy to avoid. But there have been efforts to avoid this. So one team developed a retrieval technique that involved encapsulating each DNA file into a small silica particle. So this way you can physically divide the data files into little capsules of DNA. And then another huge benefit of this is that you can label each silica ball. Yeah, you can label it with DNA so that it acts like a barcode of the DNA file. So if you want to find that specific file, you just make a complementary sequence and it can retrieve that file. And another thing you can do is you can label it fluorescently, and then you can do fluorescent particle sorting, and you can easily identify the fluorescent files you need. And then an additional thing you could do is you can search for data files with Boolean commands. So you could say, like, I want this and this, or this specific file. And yeah, I think this is moving towards the idea that eventually we can have these huge libraries of DNA files and that are labeled with DNA sequences and fluorescently. And if you have like multiple labels on one DNA file, then there's a larger number of potential labels you could have for all your DNA files. So it's working towards actually putting this to use for large amounts of data. Yeah, so I mentioned that I'll talk more about how data is actually read. So this is done by a method that is very widely used called polymerase chain reaction. And this amplifies DNA to produce millions and billions of copies, which can then be studied in greater detail. Because DNA is so small, if you want to read a sequence, you can't just have one copy of it. You need to amplify it millions of times. So polymerase chain reaction, or PCR, involves exposing DNA to different enzymes and then cycles of heating and cooling for different temperature-dependent reactions to occur. So first you apply a high temperature, like 94 to 98 degrees Celsius, to break apart the DNA double helix. Then you add DNA primers that bind to complementary targeted sequences in DNA, and you lower the temperature to 50 or 65 degrees Celsius to allow the binding of these primers. And then you add a polymerase, which is an enzyme that catalyzes the main reaction, and you raise the temperature again to 75 to 80 degrees Celsius, depending on the polymerase type. And the DNA polymerase synthesizes a new DNA strand that is complementary to the single strands. And this is the elongation step. So the cycle of denaturation of the double helix, then primer binding and elongation is repeated several times, and you can amplify the DNA to produce millions to billions of copies. So it's technology like this that really allows this DNA data storage. Some of the problems with this, though, is that the primers can crosstalk with off-target DNA sequences, and it can lead to the wrong DNA data files being pulled out and also artifacts being made. And PCR also requires enzymes that irretrievably consume some of the DNA. Like I mentioned, you need these extra sequences appended to the DNA so that it doesn't eat it up, basically. So to overcome this really recent study in 2023 by a group in the Netherlands, they used these thermoresponsive microcapsules for repeated random multiplexed data access. And I thought this was really cool. It reminded me of our biomaterials episode, because instead of like the silica beads for separating the DNA, they used natural temperature-sensitive properties of these proteins to create these capsules. So they're called proteinsomes, and proteinsomes have temperature-controlled membrane permeability. So this group previously, they used water and oil droplets to separate DNA templates into little droplets, but it was a very elaborate workflow, and it required large quantities of solvents to form these emulsions, and it was very time consuming, so it's not attractive for large scale applications. The new thermo-confined method, though, uses these proteinsomes that are semi-permeable to DNA and enzymes. So at room temperature, the membrane permeability is really high. So they can take in DNA and they can take in enzymes. But then in a high temperature, the DNA remains inside the proteinsomes and the membrane permeability is low. So the things in the solution don't mix. And so you can basically do PCR on these individual data files separately because PCR temperatures, like I said, it was 50 to 90 degrees Celsius. So at these high temperatures, the membrane permeability is, and they managed to simultaneously amplify 25 DNA files that totaled over 1.5 million unique sequences, which was much better than just bulk amplification in terms of sequence representation. And then they also labeled the membranes fluorescently, like with the silica beads, and they could apply fluorescent sorting and retrieve and amplify sequences based on that metadata of like the fluorescent label, the DNA label, and execute Boolean file searches, like I mentioned. So new technology is constantly being made to improve the ability to work with DNA and encode data and DNA. And I think it's a really promising method for the future of data storage.
Yeah, that's super interesting. And yeah, it's so important to highlight the problems with PCR. And it's so great that there's new groups and technologies trying to overcome these problems by having different methods and different techniques. I also saw another study about how a group in UCSF and Harvard developed a retrocasecorder that records the order of gene expression in cells. And basically, it uses RNA barcodes that are converted into DNA and then inserted in the cell's genome. And then they're added in a one-way manner, and they can be used to reconstruct the timing of gene activity. And then using specific RNA molecular tags, they introduce things called end spacers, which basically are a background signal, and they mark the timings of the recording of the mechanism. So essentially, it times the gene activities. And because it's a background signal, it doesn't interfere with the expression of the gene. It just serves as like a flag in order to record how far along in the gene expression process it is. And it allows you to keep tabs in terms of reading the DNA. And this reminded me a little bit of our episode with Jay in terms of having these protocols and these systems in place that just act as flags in order to double check that everything is being recorded appropriately. Like in Jay's experiments, they did these high throughput electrophysiology spike recordings over hours and hours. And they also had video data on top of it. And they had to make sure that the video data was aligned to the electrophysiology data, because timing is extremely important in that situation. And so they introduced code that would basically allow you to check whether it was recording the data correctly. And this is the same thing that they did using this retrocasecorder, introducing these tags, these RNA molecular tags. They were able to then keep track of the timing of the activity.
Yeah, that's really interesting and useful as well. It's kind of crazy how much we can do with it, like sequencing wise and mutating, but you really don't know exactly what you're doing a lot of the time. And the only way is to amplify it again, which can have errors as well, and then look at it. So yeah, when you're working with something at that small of a scale, I think it's really important to have these tags, making sure you're doing what you actually want to achieve.
Yeah, exactly. And there was another study that I read by the Molecular Information Systems Lab at the University of Washington, and they're collaborating with Microsoft in order to try and create this DNA storage. And as we know with science, you first need the fundamentals before you can spin it out onto a startup and have higher applications. So the study, I found it super interesting. They designed stimulus responsive two-state hinge proteins, which basically proteins have a specific conformation and they have a specific function. However, it would be desirable if they could behave more as like molecular motifs and be more responsive to their environment and be more adaptive. And so what they did is they developed these proteins that have a hinge-like design where basically they have an energy landscape with two low energy but structurally distinct conformations that are modulated by external inputs. So basically they can have two different shapes depending on whether or not they are activated or depending on whether they interact with a specific stimuli in their environment. And it hasn't been done before. It's extremely difficult to be able to design a protein that has this hinge-like ability of being able to reversibly switch between two different structural conformations. And now you may be thinking, okay, this is very interesting, but why is it relevant? So in terms of DNA storage, this is all very biological. And so computers have done very well, but now we also need to have better biological designs, as we've mentioned, in order to be able to read the DNA and amplify it and extract the files, etc. And so where this types of hinge proteins come to play is that they can act like transistors in electronic circuits, because they design these two-state switches, which can be coupled to external outputs and to the internal outputs, creating this sensing device. And then, for example, a normal transistor is a miniature semiconductor which regulates or controls the current or voltage flow, and it can amplify it or decrease it. And so if we have these hinge proteins, depending on how fast or slow we need to read the data, or simply being able to interact with the data in a way that's not disruptive, is very beneficial.
So would they be used as, what would they be used as specifically?
So you can think of this more as like a molecular transistor, I would say. Given its two states conformation and given that it's reversible, you can use it to interact, give it instructions or interact with it, and it's able to respond in a way that normal proteins can't, because proteins just have the same conformation throughout their whole lifetime unless they go post-transcriptional modifications. And this has been likened to a transistor in that it can regulate voltage or flow, but in this case, it's more about regulating, I guess, information. And this was developed by the same lab that's involved with Microsoft trying to develop these data storaging techniques. I'm not 100% sure what the immediate application would be, but this is definitely like a really important molecular tool in order to then be able to store DNA better.
Yeah, okay. I was just wondering, as you said, like transistors regulate the flow of current or voltage. So what is this regulating the flow of? But it's cool that it's, again, the idea of biomimetics and biomaterials, like using natural properties of biology and then like optimizing them to our own use. And I think it would be awesome if we could have like the whole DNA data storage system is purely biologically based. And the ultimate thing is that they're more sustainable. Like it doesn't require energy to be stored. It doesn't require a lot of space. So if we could make like every other step in the pipeline also biological, like the sequencing and reading and the flow of the information and everything, that would be so cool.
Yeah, definitely. And just to clarify why it's similar to transistors, it's not so much about the flow of current or voltage, but about the structural similarities, in the sense that they're both two-state switches that can be coupled to both output and internal sensing devices. So that it's like the molecular version of that. But in terms of what it would gate, I don't know. And as you mentioned, in terms of data storage, one of the main challenges is scalability. And, PCRs can only go so far. And all of these different tools open up the possibility of creating technologies that can be scalable.
Yeah, but the fact that it's so information dense means it's not a theoretical challenge.
Yeah, exactly.
I just wanted to end with mentioning that thinking about this topic has also made me reflect on like how much information is being stored within an organism because of how much information can be stored within DNA itself. Yeah, if we have eventually these like DNA data stores, then we will also change our perception of ourselves. Like we are computers, but in like the opposite way of how we normally think of it, because we normally think of computers as not being natural. But in the future, maybe the computers, their transistors will be biological, their data stores will be biological, and we will turn computers into living organisms.
Yeah, really interesting perspective.
We'll just have to wait and see.
Mm-hmm.
So we'll end this episode there. Let us know your thoughts on any other ways that data could be stored in the future and what you think of DNA data storage. I hope you enjoyed the episode.
Feel free to share this episode with your friends and family on Instagram, Twitter. Thanks for listening.